What is cell culture and why should you care? The COVID-19 vaccine that went into your arm. Have you ever wondered where it was actually made? Not in a factory, not by a machine, but inside a living cell. Cell culture is the hidden engine behind nearly every modern medical breakthrough, from vaccines to cancer therapies, even the future of food. And today, we're starting from the very beginning. Let's break it down. Cell culture simply means growing living cells outside the body, in a controlled environment. Think about your own cells. Inside your body, they live in perfect conditions. They get nutrients, oxygen, warmth, and constant signals from neighboring cells. Now imagine recreating all of that outside the body. That's cell culture. You're essentially building a hotel for cells. And you are the hotel manager. You control everything: the temperature, the food, the cleanliness, the environment. Get it right, and cells thrive. Get it wrong, and everything dies. Types of cell culture. There are 5 major types of cell culture, and each plays a critical role in science and medicine. Mammalian cells, human, animal, these are the closest to human biology. They're used for drug testing, vaccine development, and cancer research. Plant cells, used in agriculture and biotechnology, especially for producing valuable compounds like medicines and flavors. Microbial cells, bacteria, these tiny organisms power antibiotic production, fermentation processes, and even gut microbiome research. Yeast cells, a favorite in genetics research, and surprisingly, they're also used to produce insulin and in brewing. Viral cultures. Viruses need host cells to survive, so culturing them is essential for vaccine development and virology research. Real-world applications. Cell culture isn't just theory, it's everywhere. It helps produce insulin for people with type 1 diabetes. It enables monoclonal antibodies used in cancer therapy. It's behind lab-grown meat, reducing reliance on animal farming. It supports plant propagation for agriculture. And yes, it made COVID-19 vaccines possible. Lab demo walkthrough. Let's step inside a real cell culture lab. Every tool here has a purpose. Biosafety cabinet. This is your sterile workspace. It protects both you and your cells from contamination. CO2 incubator. This maintains the perfect temperature, humidity, and carbon dioxide levels for cells to grow. Inverted microscope. Used to observe cells in culture dishes without disturbing them. Centrifuge. Spins samples at high speed to separate cells from liquids. Liquid nitrogen tank. Stores cells at ultra-low temperatures, essentially freezing them in time. This is the life cycle of cultured cells, from storage to experimentation. Here's a mistake almost every beginner makes: opening media bottles outside the biosafety cabinet. It seems harmless, but within 48 hours, contamination, bacteria, fungi, everything takes over. The fix? Simple. Anything that touches your cells must happen inside the cabinet. Always. At MusBResearch, these same principles are applied in cutting-edge work. Biological scientists working in lab cultures gut microbiome cells under specialized anaerobic conditions, environments without oxygen. But the fundamentals remain the same: sterility, proper nutrients, controlled conditions. Whether you're growing human neurons or gut bacteria, cell culture is the foundation. Let's recap. Cell culture recreates a cell's natural environment in a dish. There are 5 major types: mammalian, plant, microbial, yeast, and viral. And it powers everything from vaccines, to food innovation, to microbiome science. And this is just the beginning. In the next episode, your lab is a battlefield, and we'll show you how to set it up and survive. If you enjoyed this video, found the topic helpful, or believe this content could be useful to others, please take a moment to like, share, and leave a comment below. And don't forget to subscribe to the channel for more videos like this. Thank you for watching.